Welcome back, guys, to Biochemistry. I'm glad you found your way back here. It's great to be back for another video. We're going to continue on the topic of acids and bases in the biochemical concepts. And we're going to start out with something we started learning about last time. It's the Bronsted acids and bases. So Bronsted acids and bases. So we can just use one equation to kind of sum up everything. Say we have some hydro acid and it dissolves or it dissociates to get hydrogen and its conjugate base. So this over here is a proton donor. And what does proton donor mean? That means that it's a Bronsted acid. So, any Bronsted acid, I'll just define that over here. Bronsted, I didn't spell that correctly at all. Bronsted acids donate protons. And that is something you can take for granted. Okay? And while, on the other hand, this conjugate base, why is it a conjugate base? Because we lost the H plus, so now this is basic. Conjugate base is now a proton acceptor. It can accept one proton. Yep, right? So that would make it a Bronsted base. So now say I had some other equation. That's not a good color, that's too dark. Which had H plus as a reactant, and we're adding H ions, we see that the H ion, hydrogen ion, is nothing but a proton, which would mean that when we add these together, I just realized you can't see this. When you add these together, you'll end up seeing that the A minus will accept the proton and form HA. All right, perfect. Moving my screen back there. And now, let's talk about some specifics. So an important one in the body is HCl. So HCl is a strong acid. This tends to completely dissociate into H plus and Cl minus, so the hydrogen and chlorine ions, anion and cation, in the presence of water. In the presence of water. Now on the other hand, we have acetic acid. And for all the organic chemistry buffs out there, if I'm not wrong, it's CH3COOH. That is the formula. But it's a weak acid. And obviously there are some reasons why it's weak. And it only partially dissociates. So that's the thing with weak acids. It always only partially dissociates, which means when like for example, the equation that I've written up here, we see that all of the HA goes to H plus and A minus. But oftentimes, there will still remain a small part that is HA if HA is a weak acid. Alright, so weak acid, it partially dissociates into nothing but H plus and CH3COO minus. Okay, sweet. So now that we have that down, we can talk about one of the most, if not the most, confusing topics in all of chemistry, whether that be in general chemistry or organic chemistry or biochemistry. Buffers is just something that blows my mind. It's just kind of difficult to understand at first. Now I'll just try to do this as easy as possible. In a biological setting, you see functional groups that are serving as weak acids. So you have organic carboxyls, aminos, like you have sulfhydryls, phenolics. All of these different organic molecules serve as buffers. And the weak acid portion, when it's combined with the conjugate base portion, it forms, like if they are present in the same, like approximately equal amounts, they form something known as a buffer. And what buffers do, or the purpose of a buffer, is to resist. It resists changes in pH. When does it resist? When there is a acid or base added to the environment, added to the system, maybe is the best word to say. Add to the system, right? So for example, if I added, say, an acid, strong acid, for example, HCl, that's what we were just talking about, and we had like a buffer in place of there, then you have, or sorry, that's a bad example actually, because HCl is a strong acid. If I had, say, acetic acid is what we're talking about, right? If I had acetic acid and then I had something like sodium acetate, which contains the weak, or like the conjugate base part, that reaction will resist the change in pH simply because there's, say, an equal amount of both of those substances. They kind of like counteract each other, and it stops it from happening. So like when OH is added, so let's say when OH minus is added, you see, like say it's added to the hydrogen ion. It essentially, okay, it's better to just write this down. Let's say I had an acid, HA, and I added OH minus. There's a reaction between this H and this OH minus, right? Because that's an acid and a conjugate base. So when this reaction occurs, we get H2O forming, and we get A minus. So this part resists that change in pH. Now on the other hand, if we're adding H plus to a buffer, it combines with the A minus to form HA. So because the added OH minus or H plus was absorbed by the buffer components, the total amount of these added ions doesn't actually go into the solution. Basically, that's why the observed changes in pH is lower than it might be in pure water. Okay, and there's another reaction or another equation we can use to kind of classify acids further almost. And that is a really important equation, and that's called the Henderson-Hasselbach. So I'll just quickly write down the name because it is a struggle to write it down sometimes. And I think I'm spelling it right. And I'm not going to actually tell you the equation. We're going to go through and actually solve for it. Say we have this happening. H plus plus A minus. Right, that is a very disconnected H plus. They're much better. That is very bad handwriting. There we go. So let's use some of our equilibrium properties from before that we had already established in the chemistry lab class. And we know that Ka, or essentially the equilibrium constant of this acid, Ka, is nothing but the products over the reactants. So we have H plus A minus over HA. Alright, that's pretty cool. we can do that. But now we're going to use some algebraic manipulation. What we're going to say is, let us solve, or let's do the pKa. Okay, we talked about pH, we talked about everything that's relating to like pH, pOH, and essentially what it is if it's like P of X, it's just negative log of X. It doesn't matter what's in that. It could be pH, it could be pOH, it could be pKa. And that's the point I'm trying to make here. Because sometimes they can represent the acid equilibrium also as a potential. So here we're going to do the same idea, and we already have this expression for Ka, so we're just going to negative log the entire freaking thing. Right? This entire thing is in the logarithm, and now we're going to use some pretty epic properties. Right? We're going to break this up. First thing we see is that we have a numerator and a denominator. It means there's division going on. So we can do negative log of H plus A minus. It's too high, right? And then since there's a division going on by the logarithmic rules we already know, we can do minus negative log of HA. That is all while being equal to pKa. Now we can expand this further. Ask how? Well, we have a product in the logarithm. So this could become negative log H plus minus log A minus. And you must be wondering, why is there a minus when I just said there's a product happening? Because this negative I just skipped a step almost, basically. Now there's a double negative here, so we can make that positive log of HA. All while being equal to pKa. Now I want you to notice something in this equation we just got. What is negative log of H plus? Duh, it's pH. So we can write that as pH, and now we just have negative log of A plus log of HA. The fun doesn't end there though. We're going to reverse pH and pKa. We're going to move them to different sides of the equation to get negative pH is equal to negative pKa. And I'm too lazy to actually write this part out again, so I'm just going to duplicate this. If I can find out how to. There we go. There we go. And now we're on the track. Now what can we do? We have a negative here. We have a negative on this side. We're going to multiply both sides by -1. pH is equal to pKa plus log A plus minus log HA. Oh, not A. HA. And now we're at our final step to finding the Henderson-Hasselbach equation. There's a logarithm here and there's a logarithm here connected separately by a subtraction, which means we could combine them using division. We get pH is equal to pKa plus logarithm of A plus over HA. And boom, we just got the Henderson-Hasselbach equation. Now this equation is really, really helpful when you're working with weak acids. You can even rewrite this and derive it yourself for when you're working with a weak base as well. So essentially what it's saying is this log A minus, this is, have I been writing it as A plus? It should be, oops. Okay, there's a small mistake here. This should be minus. Whoopsie daisies. Obviously. So essentially what this is saying is we're going to take the pH and the pKa. It's always going to be same, right? pKa is always going to be the same for a certain acid equilibrium. And we're going to add it to, this is where you see more of a variability, and that's where the beauty of the equation comes. You can use this in any equation you actually get, any reaction you get. So A minus is the conjugate base in this case, and HA, where this is the undissociated acid. This is the dissociated acid. So I want to go back to where we had said that we have some HA left over. Right? That's where it comes from. That's where the undissociated acid is. That's what it is. That's what we're talking about. And how do we find that? Use ice charts. If you don't know what ice charts are, go on to any chemistry website, learn about those. They're the most useful thing that will get you through any chemistry class. All right. So now we have solved for the Henderson-Hasselbach equation. What else can we do? Well, we can further use this equation to draw some titration curves. Let's talk about acetic acid's titration curve. Right? I'm just going to write that equation out again, that Henderson-Hasselbach. pH is equal to pKa plus log. And I'm going to write this out specifically for acetic acid. CH3COO minus over CH3COOH. Right, so that's the equation for this. And we're just going to draw a titration curve, which is nothing but it represents like the response of a buffer system in the addition of a hydroxyl ion. Right, so we're going to see something that's just drawing out the axes. Going to see something like this. Boom. And this spot over here, that's not the exact spot. And this spot over here, where it's tangent to the line of, x is equal to 0 and y is equal to something. What is this place? This midpoint is where there's equal amounts of this value and this value. And why? How can we determine that? How do we know that's true? Because if there's equal amounts of both of these, well, log of 1 is just 0, and we have pH is equal to pKa. So this is a line where pH is equal to pKa. Look at that. The halfway point also represents that the pH at which the best buffering is able to occur. So this is where the best buffering occurs. All right. The curve also shows that best buffering occurs at pH is equal to pKa. Good buffering occurs within 1 pH unit of the buffer's pKa. All right. So I hope that video was helpful to you in any sort of way, whether that be for biochemistry or regular chemistry. Just going over some of the main ideas again. We see the Henderson-Hasselbach equation connecting the ideas of weak acids, weak bases, Arrhenius acid bases to buffers, and then finally using that to draw out some titration curves. There's obviously a lot of practice on titration curves that are available on the internet, so definitely go and check those out, and hopefully I'll be posting some more of this stuff on my website, including some more detail on titration curves. So stay tuned for that. I'm glad you were able to watch this video. Stay tuned for the next one.